Hello and welcome to the WeXtree Academy Online. My name is Chris and I'll be your tutor for today. In our previous video we looked at questions 1 through 5 of the multiple choice paper, and today we'll look at questions 6 through 10 for the AS Biology Winter 2019 paper. Okay. Question 6. The diagram shows the monosaccharide xylose. Many xylose monomers can be joined together by glycosidic bonds to form a polysaccharide which is found in plant cell walls. Which diagram shows the formation of a glycosidic bond between two xylose monomers? Okay, well, if you remember that the combination of two monomers would be a result of a condensation reaction. Okay, and a condensation reaction would effectively create water as a product, so it has to be expelled out of here. So H2O, it's important here because this will help you eliminate the answers. As you can see here, there's H and H here, and if you go together, that would not combine to form water, so option A is not possible. The same as option C, okay, H and H does not actually go together here, it's not possible. You could think maybe B because you've got H and OH here, but if this happened, there would be nothing connecting the two of these together. There would have to be the bonding forming here, but there's already only a bond occurring briefly here, so it's not going to work. This is not possible. The only option here is between this OH group and that OH group, and when that water is expelled here, this forms a bond between the two monomers to create the polysaccharide chain. So the only option here is D, but it's very important you remember this condensation reaction and the product. It would really help you understand this question. Question 7, which color indicates the lowest concentration of reducing sugar present in a solution after testing with Benedict's? Okay, well, this is an easy question you should know from GCSE. If you remember the various colors, so blue is a negative result, followed by green, which is a little, like very little, followed by yellow, followed by orange, and then red. So this is your highest, and this would be your lowest. Okay, so that would be negative actually there. So here, well, brown, well, that's irrelevant. It's not a color on the scale. However, green is your lowest concentration after a negative result. Red is a very high concentration and yellow has more than green, so D is incorrect as well. Okay. Question 8. Which molecules are monomers? Okay, so you've got ribose, glucose, deoxyribose, and sucrose. Well, ribose is a monomer. This is a pentose sugar. Glucose is a hexose sugar as well, so that's also a monomer. Deoxyribose is also a monomer. However, sucrose is not a monomer. This is a combination of glucose plus fructose. Okay, so sucrose is not a monomer here. And as a result, the only option is A. Okay, so 4 is clearly not a monomer. And as you can see, by knowing that, it eliminates the other 3 options straight away. Okay. Question 9. Which molecules have properties that are dependent on hydrogen bonding? Okay, well, if we look here, we've got cellulose, glycogen, hemoglobin, and water. Okay, so if we look at cellulose as a structure first, well, hydrogen bonding is important here because it takes place between the hydroxyl groups, and what it does is it gives the tensile strength to the cell wall of plants. Okay, so cellulose does depend on the hydrogen bonding. Now, glycogen does not depend on hydrogen bonding because what happens here is generally it's the glycosidic bonds that are occurring between the different molecules of glycogen. However, the hydrogen bonding may take place between the surrounding water rather than the actual molecules of glycogen itself. Well, hemoglobin as a protein, as a quaternary structure protein, will rely on hydrogen bondings to maintain its relative structure and shape. If this functional protein has bonds altered, this could alter its functionality of transporting the oxygen, which could have significant damage. Okay, and water, well, as we know, there's lots of water molecules and they all have hydrogen bonding between each other. and therefore the water molecules require quite a detailed interaction of hydrogen bonding that occurs. So 1, 3, and 4 are all true, and they're all related and dependent on hydrogen bonds. However, here the glycosidic bond for glycogen is incorrect. So anything with option 2 in it is incorrect. Okay, option C is the correct answer. Question 10. Some features of triglycerides are listed below. Can be a liquid or a solid at room temperature, contains a high proportion of carbon to hydrogen bonds, it's insoluble in water, and less dense than water. Which of these features make triglycerides suitable for energy storers? Okay, well, if we look here, the idea of density, okay, the idea it's less dense is irrelevant, okay? Being less dense does not necessarily mean you're better at storing energy. Being a liquid or a solid, well, okay, that doesn't really matter at this point in time either. That's a physical property rather than anything useful about storing energy, okay? Now, being insoluble in water might offer some questions for you, but the fact it's insoluble means it cannot be dissolved. If it cannot be dissolved, then it cannot be metabolized at that moment in time because of the hydrophobic nature of the molecules. So if it's not going to be able to be absorbed by cells straight away, it's not going to move in and out, or it's not going to be metabolized directly in that form. Okay. And the idea of high proportion to carbon to hydrogen bonds, well, this means that when it's hydrolyzed, each bond being broken can release more energy than if it was as an individual unit. So when the triglycerides are broken down, it releases 3 fatty acids to then be metabolized further for energy release. Okay, so the only options here are 2 and 3. Okay, so anything with 4 in it, well, we could have eliminated straight away. And option 1, again, being liquid or solid, does not really make a difference at this point in time. Okay, so it's option D is the correct answer. Thanks for watching our video today. Please register on the website for a free demo, and I look forward to seeing you in our next video.